Hi, my name's Dr. Christopher James Ward, and I work at the Kansas University Medical Center. And today I want to discuss how to make monoclonal antibodies. So this talk will be in two major sessions. First we're going to talk about the theory behind monoclonal antibody production, and then second we're going to go into the lab and actually do hybridoma fusion. So the first question is, what is a monoclonal antibody? Well, basically it's one type of antibody with one particular paratope, that's an antigen binding region, which sees between 5 and 8 amino acids, or about 3 or 4 carbohydrates, depending on what the antibody is raised to. So let's have a little look at some of the theory. So when a higher vertebrate, a mammal or bird, is confronted with a foreign antigen, it produces many different antibodies that recognize the antigen. So let's consider an antigen like ovalbumin, which will inject into a mouse. The mouse has B cells with millions of different specificities. A subset of these specificities are activated by the ovalbumin, and the mouse expands B cell clones, each of which makes a particular antibody to a structure known as an epitope on the surface of the ovalbumin. So as I said, the epitopes are usually 5 to 8 amino acids long. Now, often there are multiple different B cells making antibodies to one particular epitope, and there may be hundreds of epitopes per antigen. So what you get out of the animal when you take its serum is a polyclonal antibody, because multiple clones of antibody producing cells have been activated in response to the antigen, and are making immunoglobulins against your antigen. Now, this might be well and good for some crude experiments, but the trouble is that you're limited by your animal. It'll eventually die, or in some cases you have to kill the animal and take all its blood. Even if it's a large animal like a rabbit, you'll only get about 30 mils of blood. And so eventually you have to revaccinate an animal, and you can't be sure of getting exactly the same polyclonal serum. Now, if we come back to the B cell clones that have been activated, there are many hundred different antibody types that recognize different epitopes of the antigen. And the central idea here is to produce a cell line that can make one specific type of antibody recognizing one epitope, a so-called monoclonal antibody. Unfortunately, it's not easy to isolate individual B cells and expand them to make a monospecific antibody. The conventional approach is to immortalize B cells by fusing them with a myeloma cell that confers the ability to grow forever. This is a Kohler and Milstein technique that won a Nobel Prize. So this technique then solves the problem of generating a cell line that is both immortal and also can make one particular monoclonal antibody of known specificity. So here's a schematic of what's happening. We've got two different cells that we're going to actually merge or fuse together. First is a B cell from an animal that has been vaccinated against your particular antigen. And in this case, this B cell has got antibody molecules on the surface that are cognate, that means recognize the antigen. Unfortunately, it'll die in tissue culture on its own. Here's a myeloma cell line. Now, a myeloma is a malignant tumor, B cell tumor, and this myeloma can grow forever. Now, the idea is basically to confer immortality on the B cell by fusing the two together to make a hybridoma that is also immortal. And hopefully this hybridoma will then secrete a monoclonal antibody with that reacts to your antigen. And as I said, these monoclonal antibodies can be incredibly specific. But the problem is that in your tissue culture, you will have a hybridoma and you'll have a myeloma. And the myeloma will grow quicker than the hybridoma and swamp out your hybridoma. So in this case, the myeloma is HPRT negative. So that means it's hypoxanthine guanosine phosphoribosyl transferase negative. Now, that's a big word for an enzyme that does something very simple. It takes purine precursors and scavenges them and allows them to be made into A and G. So in this case, it's hypoxanthine that will supply, and the enzyme is part of a pathway that will make first inosine and then A and G. And now, if you're negative for that, you are exquisitely sensitive to aminopterin, which blocks de novo synthesis of thymidine and the purines, A and G. And so you would be sensitive to this medium, hypoxanthine, aminopterin, thymidine. Hybridoma gets a good copy of the HPRT gene from the B cell, and so is resistant under HAT selection. And that's how we get rid of the myeloma. Okay. Basically, we give them a blast of HAT just at the very beginning of the protocol to kill these, and then concentrate on the hybridomas. So how do we do this? How do we select for stable fusion hybridomas? B-cell myeloma fusion is accomplished using polyethylene glycol, which is just a chemical that allows membranes to fuse together when cells are in close proximity. Or alternatively, if you've got the machine, you can use electrofusion, which uses an oscillating electric field at about 1 megahertz to line up B cells and myeloma cells, and then gives a pulse, a voltage pulse, to fuse them. We don't have such a machine, but it is a highly efficient way of accomplishing a fusion. Now, once you've done your fusion, unfused B cells will die in tissue culture, but both the myeloma cells and the hybridomas will be immortal. And so, as I said before, we must select against the myeloma cells, and to do this, we use the fact that the de novo synthesis of nucleotides A, G, and T are dependent on the presence of tetrahydrofolate, which is synthesized by the enzyme dihydrofolate reductase. And dihydrofolate reductase is inhibited by aminopterin or methotrexate folate antagonists. So while cells can survive aminopterin treatment, if we give a supply of thymidine and a source of the purine precursor for the salvage pathway, in this case hypoxanthine. Hypoxanthine is converted to inosine monophosphate, precursors of ATP and GTP, by hypoxanthine guanine phosphoribosyl transferase. So the myeloma cell is HPRT negative and so dies rapidly in the presence of hypoxanthine, thymidine, and aminopterin, as it can't use the salvage pathway, and the de novo pathway is blocked with aminopterin. The hybridoma has a functional HPRT gene from the B cell and so can survive in HAT. The myeloma confers immortality, which the B cell lacks. And so the only survivor under these conditions is a hybridoma. So in summary, the myeloma has intact de novo nucleotide synthesis but lacks a salvage pathway. The myeloma fusion partner is exquisitely sensitive to aminopterin. It is immortal and can confer immortality to B cells. The B cell has both salvage and de novo pathways but dies in tissue culture by apoptosis. It's resistant to aminopterin in the presence of thymidine and hypoxanthine. The hybridoma is resistant to HAT medium and is immortal, and so it's the only cell that will survive. Okay, so that's basically what we're going to be doing. So what do we need? If you're going to do this, we have to order IMDM, which is a basal mouse cell line medium. Then we need hybridoma qualified fetal calf serum or fetal bovine serum. Now this has been tested to support hybridomas and myelomas, and it's also mycoplasma free, and that's important. We need pen and strep antibiotics just in case there's some contamination when we take out the spleen. We need glutamax, or sort of glutamine. We need non-essential amino acids, hypoxanthine and thymidine, HT. Now this just helps support hybridomas after selection. And the actual selection medium is hypoxanthine, aminopterin, thymidine, and we get that in a 50 times stock solution. And we also need adenine, and I'll tell you why. Although the scavenger pathway can make A and G, we add adenine, and it's just been observed that this makes selection a little bit less harsh. We're still selecting on the inability to make G, so the myeloma can't make G under these conditions, but we do supply A. It just gives hybridomas a little bit better chance of survival because they can make G. So what we do is we dissolve 258 milligrams of adenine HCl in approximately 80 mils of deionized water. And then we buffer it if necessary to dissolve any crystals. We buffer it with 5 molar sodium hydroxide. Then we bring the final volume to 100 mils with deionized water. We filter, sterilize and store in 6 mil aliquots at -20. So that's important. Then to make the complete medium, we use 450 mils of IMDM, 50 mils of fetal bovine serum, 5 mils of pen and strep, 5 mils of glutamax, 5 mils of non-essential amino acids, and we filter sterilize. To make the selective medium, the AHAT, we add to the complete medium 2.7 mils of adenine and 10 mils of HAT 50 times. And then to make the supportive medium, non-selective, which is called AHT, we add 2.7 mils of adenine to 500 mils of complete medium, and then add 5 mils of HT to that. Okay, and so that's a supportive medium that we use when we're taking the cells out of selection from the AHAT to AHT. The HT is there to really support the hybridomas as they come out of selection. Most of the myelomas will be dead by that stage. So the vaccination protocol. Now this whole protocol was basically given to me by Tom Beto at the Mayo Clinic, and I've modified it somewhat. We work on a human disease, autosomal dominant polycystic kidney, and we raise mouse monoclonal antibodies against human proteins. And this is because it's a human disease. We want to probe polycystin function in humans. And two, the mouse is phylogenetically quite far away from humans. It's about 98 million years. So that's a Cenomanian period, which is the lower part of the Upper Cretaceous. So our ancestors back then, our last common ancestor was back then when dinosaurs ruled the earth, basically. So in this case, we are making antibody to the top domain of polycystin 2. There are 2 existent monoclonals to PC2, YC2 and D3, but they're both cognate, or they recognize the intracellular C-terminal tail of the molecule, and we wish to make monoclonals to the extracellular top domain of polycystin 2. So the proteins are made in E. coli, and there'll be a talk on that, as His6 tagged proteins, which are purified by cobalt affinity chromatography under denaturing conditions, and then dialyzed against PBS to induce precipitation. And this precipitant is actually a good immunogen, and it's used as the antigen. We mix it with Freund's complete adjuvant FCA, and the suspension of aggregated protein is mixed with this. And then we inject 200 micrograms of protein in 100 micrograms of FCA subcutaneously at the base of the tail. We give 2 injections, and we use 10 mice. And we only use Freund's once. You're not allowed to use it more than once, and that's important. 3 weeks later, we inject 200 micrograms of precipitant protein subcutaneously, and then we repeat that 3 weeks later, and then 3 weeks after that. So you're going boost, boost, boost. And then we take 50 mils of blood from the tail vein for assay. The plasma or the serum is assayed by Western blot. In this case, the target is full-length PC2 TAP. The TAP is a tandem affinity tag, and it has a flag tag, a triple flag tag, and 2 strip tags. And this is expressed in Chinese hamster ovary cells. The mouse that makes the best antibody response is selected and given an intraperitoneal injection of 100 micrograms of protein on each of 3 consecutive days, and then the spleen is harvested on the 4th day, and I'll show that in the practical. So to harvest the spleen cells, the mouse is killed using CO2 asphyxiation. This is the kindest, gentlest way and less stressful way of actually killing a mouse, followed by cervical dislocation. The mouse is then immersed in 70% ethanol with providone iodine to sterilize the skin. The spleen is removed using a sterile technique. The spleen is then cut into pieces and passed through a metal mesh in the presence of warm IMDM medium. Cells are recovered by centrifugation at 3000 Gs for 10 minutes. Then you have a kind of red pellet which is resuspended, and the red cells are then lysed in 5 mils of ACK medium for 1 minute. The lysis is halted using 8 mils of PBS, and the cells are recovered again at 3000 Gs for 10 minutes. The cells are resuspended in the medium of choice. Now you'll need 2 mice to supply spleen feeder cells that are just put into complete medium. Okay. And then you'll also need to get the spleen out and make lymphocytes from your vaccinated mouse. So 3 mice in total here. You also need your fusion partner, which is our FOXNY myeloma cells that are grown in complete medium. Cell cultures are selected that are growing rapidly and have about 500,000 cells per mil. Cells are recovered by centrifugation at 1000 Gs for 5 minutes, resuspended in PBS, recovered by centrifugation as above, and resuspended in warm IMDM medium with no additives. Now the idea is just to get rid of the fetal bovine serum here and have them in a serum-free medium. And you also get splenocytes from your vaccinated mouse using the previous protocol. So the immunized splenocytes in IMDM and the FOXNY cells, again in IMDM, are counted and mixed in a ratio of 1 FOXNY cell to 2 splenocytes. The mix is then pelleted by centrifugation at 1000 Gs for 5 minutes. All the medium is removed and the pellet is disrupted. You then place a tube in a bead bath at 37 degrees and add 1 ml of PEG 1500 dropwise over 1 minute. You incubate for 70 seconds at 37 degrees with agitation, and then you add 1 ml of warm IMDM drop by drop for 1 minute, then 3 mils drop by drop over 1 minute, then 11 mils of drop by drop for 1 minute, and then centrifuge very gently 500 Gs for 5 minutes. Then you recover the pellet and gently resuspend it and add it to the 100 ml complete medium with feeder cells from 2 mouse spleens. So that in this 100 mils you've got the feeder cells from 2 spleens, and you've also got your fusion mix, and it's very fragile at the moment. You want to then plate 100 microliters per well in about 10 96 well plates. So if you're using tips to do this, you cut the tips and make the orifice wider so you don't shear the cells. Then you incubate overnight in a CO2 incubator. The next day you add 100 microliters of AHAT medium to each well. Okay, and this is a selective medium. On day 3 you remove 100 microliters of the medium and add 100 microliters of AHAT with the aminopterin in it. Then on day 7 you start to remove the aminopterin. You remove 100 microliters of medium and then 100 microliters of AHT, and then you repeat that on day 10. And on day 11 and to 14 you're ready to start screening your cells. So let's get into the lab and actually perform this experiment, and I'll show you how to do it in practical terms. So here's hoping we've sterilized the whole outside of the mouse as much as possible.
always make sure that there's no feces or anything like that associated with the mouse, because that can be a problem. And so you've got 2 dead mice. They're biopsies, which are syngeneic with the cells, with the mice we vaccinated. So there's no mixed lymphocyte reaction or anything like that going on. So here we have 2 mice. I'm getting there close. Okay. What we're going to do is we're going to go do unsterile first, just opening them up and exposing the peritoneum. Okay, and then we're going to change the tools and go in and open the peritoneum and fish out the spleen and put it in there. Okay, and then we're going to grind up the spleen. The first thing to do is just open up the mouse like that. That's the way I do it. And try not to get down to the peritoneum. Don't open the peritoneum, whatever you do. So there's a peritoneum there, that sort of sac that you see, and it should be kind of sterile. Okay, and the spleen, right, is on the left side of the mouse, right up here. But do the same for the other one. So just tear the skin back. Really important that everything is kind of sterile, because if it's not, and you get bugs in it, it's the end of your experiment and it won't work. You try to expose the spleen. You see, there's the spleen right there. Do the same on this side. Right. Okay, right. We forget about all this now. What we're going to do is new sterile tools, new gloves. Keep your fingers back from it as much as possible. And first of all, just kind of something like this. And then what I'm going to try and do is get the spleen out as sterilely as I can. Grab the peritoneum, cut, and cut towards the spleen. And grab the spleen. Again, cut. Just cut in there. Done. One spleen. I didn't touch anything there, so that's good. Try the other one. Grab a bit of peritoneum. Okay. Cut towards the spleen. Grab the spleen. Just try to tease it. Basically, I think it's pancreas actually on the spleen. Yeah, tease it up. Do it as cleanly as you can, alright? So there you have it. You got 2 spleens, 2 mice that you don't need anymore, so we'll just pop those to the side. Now then, what we're going to do is we're going to get ourselves a nice sterile spatula. So of course my hands aren't, they're clean but they're not sterile, so you got to remember that. And we're just going to have this. And here you've got to get quite close to the blade. Alright. So the blade is sterile. Well, at least the very tip of it is. And then what we're going to do is going to chop up the spleens as much as we can. Okay. So I'll have to use this again, just chop it up. Just anything that comes in contact with the tissue has got to be kind of no bugs and no U. Okay. Just really important. Let's chop it all up. Get them together now. Yeah, we might be able to reuse this guy if it survives the storm. So then what we want to do is just take this like this. Pick up your spoon. It's kind of drying out there. Flood this with flood this by MDM. We'll probably have to re-autoclave that guy. Grab it. Just push the tissue through. As much as you can. Push it through. Okay. So there we go. I've done that, okay? So these are the feeder cells. This, and this. And what we're going to do is we're going to take this and spin it down, and then we're going to get rid of the red cells by using ammonium chloride PBS lysis of red cells, and that'll leave the white cells intact. So we've got a bit, so we better do that now. For that, I'm going to use Asset. Have to get that autoclave, even if you have to run the autoclave for itself. Right. here's hoping that this is mainly sterile. Okay. Now what we want to do, we want to spin it down, and we're going to take it up in ammonium chloride for 1 minute, and then we're going to dilute that ammonium chloride away. And what will happen is that ammonium chloride will pop the red cells, and we'll be left with the white blood cells from the spleen, which are going to be macrophages and B cells and some T cells as well. But we're really, in this case, we're using them as feeder cells to support hybridomas. And so we hope there's plenty of macrophages there, which would be good. So it looks kind of like that. So let's spin it now very quickly. I only give it 500 G for about 10 minutes. We have to allow some of this to be perfect. Right. And okay. Should just do it for us. That's good. Okay. I'm going to bash it up real good. Bash up that pellet. So this is from the spleen. And then what we're going to do is we're going to do ammonium chloride lysis for 5 minutes. So we've got some ammonium chloride here. The biggest problem you've got is actually contaminating it from yourself. The mouse has been embedded in, so it's pretty clean. Contamination is an issue. The last thing you want is a mold or even worse, bacteria growing in your hybridomas. Sorry, you can keep. Right, like that. And you just want 5 minutes. 1 minute, sorry. 5 mils of this. Okay, could you start the time? So it's going to be exactly 1 minute. That's it. Okay, 8 mils. Good. Looks good to me. Screw it back on again. Right, so what we're going to do is hopefully the lymphocytes and macrophages have all survived that, and the red cells have popped. And you've just got red cell ghosts in there. And we're going to spin it out again, and then we've got 100 mils of warm myeloma medium, and we'll pop them in there, and these will be our feeder cells, and it should be good. Now, what we've had to do is, because earlier on I had a little accident in contaminated something. I had to pop it into the autoclave again, but we'll at least we were aware that it was a mistake, and we can recover from it's part of the protocol, I suppose. So let's spin this down, and we should get a nice white colour in a little bit. Just going to start again, and then we're off. Okay.
Let's go.
Okay, so we've just finished the red cell lysis with the ACK ammonium chloride buffer, and as you can see, the pellet is now more white. It's got a little bit red on the top, and there's a lot of red up here, which is hemoglobin from the lysed red cells. Okay, so what we want to do is resuspend this into a more complete medium, and this is going to be IMDM with 10% hybridoma quality fetal calf serum. It's got glutamine, it's got amino acids, and most importantly, it's got an antibiotic cocktail, penicillin and streptomycin, which ought to kill most things. As you can see, we're trying to do this as sterilely as possible, but sometimes, bugs do creep in there, and they either come from you because you've got skin exposed, or from your mouse because it's not externally kind of cleaned up. For example, there could be fecal contamination on the skin or something like that. So you've got to give them some time, the mice some time in the povidone iodine to really sterilize the outside. As you can see, this is nice and white now. Okay, so I'm going to I'm going to decant off supinate and then resuspend it. So I don't use a sucker or anything, I just pour it quickly as I can. And then, now you've got this at the bottom. I'm just going to show you, just flick it. Just flick it. I'm trying to break it up a bit. Sometimes you've got to be a bit rough with it. But if you just put the media straight on top of it, you'll just get a big clump of cells, so you've got to break up the pellet. That's really very important. Kind of learned that the hard way, nobody taught me, but here you go. Just keep doing that. Keep going, keep going, keep going. It's not quite there yet. Something's got to be a bit rough with it. Yeah, and there's little bits and pieces floating around. Oh, there is here. Let me just bash that a bit. Okay, that'll do. Right, so let's take some of this good medium here, myeloma, no medium myeloma medium bottle here. So IMDM, 10% fecal calf, penstrep, glutamine, non-essential amino acids, all the goodies. Everything a cell could possibly want. No selection at this time, okay? That's important. Let's hope we don't get any clumps, but sometimes you do. I'll just be gentle here now. Let's just have a quick look at it outside. Let's just squeeze it up. Okay, so we've got a bit of gubbins at the bottom, so we'll just let that fall out because we can't really use that. It's probably just a piece of spleen that hasn't quite just been disaggregated. So, right, got that like that. We're going to take the top bit, okay, and put it in here, okay. There's probably 50 million cells in there, something like that. We can count them if necessary. Beautiful. There you go. There's your feeder cells. Okay. Still a little bit of debris in there, but you know, that's life. Now we just need to write feeders on these. And so we'll just leave these floating around. And what we'll do is these will feed the hybridomas. So the next step is to do the hybridoma bit. And so we've got a vaccinated mouse that's coming. And again, we'll do the same thing. We'll take its spleen out and disaggregate it. And lyse the red cells and then get the lymphocytes. And then what we'll do is then fuse them with the hybridoma. We've got hybridoma growing in the incubator at the moment. It looks really good. Okay, so there you have it. I'm going to just have a quick look at this under the microscope, just to see what the cells are. Get the spleen out of a mouse again, and then we're going to mash it up and get the spleen cells out. Mouse. First things first, we're just going to open the skin. And then we're going to change gloves. And then we're going to open the peritoneum and get the spleen out. So we just open this. Oh, there's a big spleen there. So this mouse has been vaccinated against polycystin 2, the top domain. and it looks like that spleen is huge. So that's a good sign. Okay. So I'll just point it out to you. I'm gonna go for that big juicy spleen. Looks good actually. It's really good. When you get one like this, you usually get a claw. It's much bigger than the wild, the unvaccinated spleens that we're looking at before. So I'll just grab that spleen up there. Look at how friable it was. Yeah, you see that? Really friable. There. So then we have that like that. Put spleen out of there. Hope I didn't touch anything. I didn't open any bit. Again, we got together. There's a scaffold again. And then so that's a big spleen. Really, really long. It's a good sign. I'm going to dice it up a bit. It's better if I took it out. Cool. Sterile. I'm actually going to hoover up this stuff here and use it so I don't lose any cells. And I'll just pop this up a little bit now. Put this back in there. So it's a bit of a guddle, but we'll get there. This question is just getting nice disaggregated spleen cells. There's a lot of stuff to throw away in there. Okay. So what I'm going to do is just mash these through. Now the big thing is I just pray that I don't get any bugs growing. So just crush this through. See, it's a very friable spleen. You know, if someone's got Epstein-Barr infection or something like that, and splenomegaly or myeloid leukemia or something like that, then this spleen can really be damaged quite easily. You've got to be very careful about that. If someone does have splenomegaly, usually for humans, for it to be dangerous in humans or to be palpable, it's got to be 3 times bigger than normal. Now this mouse spleen was pretty big. It's about maybe 2 or 3 times bigger than normal, so that implies it's been making an immune response against our antigen. So we injected it in full Freud's adjuvant influenza antigen, and then we gave it 2 boosters, and then we checked, and this one was making antibodies. Then we give it 3 quick boosts in a row on day 1, day 2, day 3, and now this is day 4. We've just taken it, so it should have what we're looking for. Plenty of B cells that are recognizing or cognate to the top domain of PC2. So I just hope we get something. To get our hybridoma cells ready, and they're FOXNY, okay, and they are HPRT negative, okay, and so these are growing up quite beautifully, and what we've got to do is spin them down, put them into PBS, then put them into IMDM with no antibiotics, okay, or anything like that, and then count them basically, because the ratio of B cells, hybridoma cells, is actually quite critical. So we'll have to go upstairs to measure the number of cells. We've actually got a cell counter up there that's pretty accurate. But once we've washed them and got them into the IMDM, then to the IMDM. Sorry. So these are FOXNY cells. I got these from Tom Beto at the Mayo Clinic. A really nice fusion partner. And basically this is a modification of his protocol. Okay.
Gonna have it on Monday, so
Okay.
thank you.
Good, good. Thank you very much. So we want to just give these a gentle spin. This will only be about 5 minutes. Right, okay, you can see these are spleen cells, and we have to lyse them. Okay, but they're bright red. So let's get back onto this again. So we're multitasking. We're spinning down our FOXNYs at the moment. And we've got our immunized splenocytes here. Turn that off. Okay, remember, break it up real good. Real good. And then we need 1 minute timing. So we'll have to time 1 minute here. Okay, thanks, man. See, so this is being a bit of a pain. Oh yeah, it's better now. Good. Good. So again, this was a very big friable spleen. It's a good sign. As long as it doesn't get bugged, we ought to get a PC2 monoclonal. So this will be a mouse, hopefully anti-human. The reason we're doing this is we ran into problems with the 2 monoclonals that are available, which are D3 and YCE2. They're both at the C-terminal tail of polycystin 2, and I'm very suspicious that there's something happening there, that it's been cleaved off, because I can't see it in a place where it should be. I think it should be at the focal adhesion. I can't see it with conventional PC2 antibodies. So I think there's a modification there, perhaps a cleavage. Certainly I've seen a cleavage in the exosomes. So I want to make one to the top domain. That'll be quite tricky because it's glycosylated, but you know, it's life. 5 minutes. Approximately 5 minutes. There you go. Let's move on. Okay, could you start the timer?
Yep.
Just in case we've got a binding problem or something. So it's quite a long day to do hybridoma. Trying to do it properly, it's quite stressful. So it's worth taking a break at the end of it. Alright, so just wait a minute, and then what we've got to do is, of course, we've got to stop that reaction, as they call it, just by topping it up with PBS.
10 seconds left.
10 seconds, thank you.
And the other one's 20 seconds behind.
Okay, got you. Just tell me when that turns up.
How's that?
Okay, I'm on it. So let's go to the tank. There we go. Oh yeah, I think that worked now. Yeah, good. And then the other one. So these myeloma cells are big cells that come down quite easily. So we can just stop in a minute. Yeah, beautiful. Good. Alright, so there we go. So yeah, that's good. That's good. They both look pretty good there actually. There are some bitties, but we'll get rid of them. It's not too big, not too small. That'll be enough for them to do this, I think. Right, so we've got these guys. So you notice the medium's not gone too yellow. Which is a good sign. You want an exhausted medium, you want them growing rapidly, really fast, and perhaps we lost some cells there. Let me see. Doesn't matter, I've got fallback position if I do, alright? We've got, yeah, so now what we're going to do is we're going to give it quite a generous wash. We'll use another centrifuge to spin them down again, and we'll take them up in IMDM about 25 mils and then count. I reckon, you know, if we get, you know, a few 100 million cells, in both, we can do the calculations and do the fusion. The fusion is a little bit tricky, but I'll show you how to do it. What we're going to do is we got 6 times 10 to the 6 cells for the lymphocytes, and for the FOXNY 4.62 times 10 to the 6 per mil. So we've got 11 mils. We've got 11 mils of the lymphocytes. So it's 11 times 6 times 10 to the 6, which is 66 times 10 to the 6 cells. Okay. And for every lymphocyte, we want half a FOXNY cell. Okay, so 2 lymphocytes for 1 FOXNY. Okay. So that means we need 33 times 10 to the 6 FOXNYs. Right, so that means 33 divided by equals, we need 7 mils of them. 7.14 mils. Okay, I'll just check that that's right. So we want more spleen cells than FOXNY cells. So if we take all of our cells, all 11 mils of them, and add 7.14 mils of FOXNY cells, and gently spin them down, we'll be ready to go. Okay, and so what I think I'll do is I'll do that in a 50mm tube with an open bottom. Okay, right, let's do that now. Top off. We'll take 7 mils of these. Some 0.1 in fact. Mix them up a bit. There we go. That'll do. Okay, now then, let's take the lymphocytes. All of them, basically, and use the same tip. So there's a lot of death down there already, just from the cells being spun down and all that. They're suspended. So there you go. There's your mix. Oh, okay. So that's our mix. Everything we need. Call it mix. So we'll give that a gentle spin down 5 minutes at about 500 Gs or something. Call it mix. Keep a hold of this in case we need it. I don't think we will. Don't need that. Don't need that. Don't need any of this. Get rid of it. Just have to tidy yourself up as you go along. Right, we're good. I'm happy with that now. Gently spun down the mixture. We've got hybridoma cells and spleen cells in there. So what we're going to do is gently tip off everything. Just get rid of it. You can get the worst of it off, okay? And then I'll leave it for a second to see if anything drips down to the bottom. You want to make sure it's kind of dry down there. There's a little bit of fluid there. I'm going to get that off, okay? Just to keep it as concentrated as possible for the peg, which is right here. So what we're using is polyethylene glycol peg. Okay. So just take the lid off again. Okay, so it's one use, okay? I suppose if you're really desperate, you can use it multiple times, but that last fluid off the bottom. Okay, just a long one like this. Okay. Right down the bottom. Want it as concentrated as possible and break up the pellet. That's the first thing to do. Just a little bit again. Right. Tricky bit. So over 1 minute. We've got to add 1 mil of this on these beads, which are 37 degrees. Try to get it off. Banging it. I'm going to be really careful, okay? Can you give me 1 minute?
Yep.
Okay. You ready? Okay, starting now. Or down the side. Oh shit, I'm going a bit too fast now. How long have I got now? Just cut.
Been 15 seconds.
15 seconds. I'm not as good as some of the old pros on this one. How long now?
25 seconds.
Okay, just accidentally put it all in. Okay, that wasn't perfect, but how are we doing?
3, 2, 1.
Okay, we're okay. I just slowly put it in over a minute. And then, could you give me 90 seconds? What's it say? Just print it. Looks good to me. Looks good to me. There you go. Warm. Fine, yeah. Keep a big volume of it because I want it to be kind of warm. Kind of. Yeah, keep it warm. Okay. How long have we got now?
Oh, time.
Is it time now?
Or 30 seconds.
30 seconds. It's alright to me. Looks like it might be working actually. If we can get a good monoclonal here like 7012 or 19 or 161 or some of the other ones we've got.
Alright, time.
Thank you. Right, you've got to give me 1 minute to put this stuff over it, okay? And I've got to be careful here now. Okay. Tell me when. Yeah.
When?
How am I doing? What time is it?
You got 40 seconds left.
40 seconds.
30.
Tell me when it's 20.
20.
That's good. Tell me when it's 10.
10.
Good.
5.
Is that it? Okay, that's one done. And then we need to do reloads. Okay. Could you give me another minute?
Yep.
Okay. Tell me when.
No.
Okay, thank you. What do you think? Tell me when you're at 45.
$45.
Good man, thanks. Tell me when you're at halfway.
$30.
Okay, got it. Keep going. Tell me when you're at 20.
Oh, 15.
50, hold on. I'll hurry up.
10. 5.
Okay, done. We're getting there. 9. diluting at the peg a little bit without killing the cells. Okay, give me a minute.
Yep.
I almost dropped it. Months of work. Just making the antigen was expensive. In this pellet, there are myeloma cells and B cells, and also hybrids, cells that fuse together. And we're going to select for those over the next week or two, and hopefully one of them will be making an antibody to our polycystin 2 top domain. Okay, so what we're going to do is take this off and put it into warm media, this stuff here, and then add it to our feeder cells, and then plate it out into 10 96 well plates, 100 microliters each. So let's do this. Okay, very gently pour this off. I like to pour. A lot of people like to suck, but I pour. And then I just gently do this. Break the pellet up a bit. Not too harsh. Not very fragile at the moment. That's the other thing about them. You don't want to draw them through a fine tip or anything like that. You just want to be as gentle as possible. Just flick them in there. And then we're going to use full medium here. No selection just yet. We'll wait a day before we start selecting. We want them to. Well, that looks alright to me. Yeah, it looks kind of alright. There's no lumps in there or anything like that. Okay. Right. Let's put about 10 mils in there. And then we'll transfer it to our feeder cells. And then we're good to plate. So here's the myeloma medium that we're talking about. And it straight through. So I don't select initially. I just have them in very rich medium. MDM with all the goodies. And antibiotics, of course. Just in case we haven't been super sterile. There we go. Just going to pipette it up. So I'm quite pleased with that. That looks good to me. Right, and into the feeders. It's a waste of a good bottle, but I can't find anything else that takes about 100. There you go. Okay. Right. Okay, just gently mix them all up. Now then, what we have to do is tidy up and get 10 plates that are round bottom plates. Let me show you what we got. Our round bottom plates. So let me open up and show you one. Look at the bottom, they're round. Okay, let's collect the cells in the bottom. That's good. So what I'm going to do is I'm just going to take 10 plates. And we're going to have to number them. And so, you know, whenever you see a monoclonal in the literature, it's always called A10, 1A10. I'll show you why. So each of the plates gets a number. Well, first of all, just so we don't mess up the lids, I do this. So you can always map them back together again. Okay, and then you have plate you're going to have plate 1, and if it's a positive clone, say it's D7, it'll be 1D7, okay? And that's where these funky names come from, if you ever wonder. So we can actually plate them out in any order. I'm just going to call them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I think I'll probably actually have 11 of these. Maybe even 12, I don't know. I'll keep one just in case, okay? Right, so, haha. We've got this far. What we need now is this is why people usually ruin the experiment, okay? You don't want to pipette them through a thin nozzle as much as possible. You want to actually cut your tips so you don't break the cells, because they're very fragile at the moment, hybridomas. Okay, so I'll show you that in a second. Another thing is you need some of these as well. This is why I carry a knife in the lab. It's a good way of getting into things sterilely. You can just do something like this. Of course. Oh, jeepers, come on. I need to sharpen this guy. I'll never take the first one. I always take the second one, just in case. Just want to be careful. Because this is the important bit. You get bugs. Ah, it's so annoying. Okay. Put it over here. Right. Here we go. So again, swirl them around. You want to be gentle with them. Mix them up. Mix them up. Mix them up. Mix them up. And then I'll put about 50 mils in. I'm going to just pour it. The first one. Right, now, look at this. I have sterile scissors here, okay? So, set this to 100. That's what we're going to use. Oh, 100. God, I'm tired. It's a tiring business. Right. There you go. So, 8. And this is an important bit. Don't forget to cut the tips. Quite wide. Ping in everywhere, we'll clean it up in a bit, okay? Right, there we go. Still sterile, kind of. I've got antibiotics in here, but start with anyone, it doesn't matter. Okay. This is where you really have to be covered up. Hi there. So we've just plated out our monoclonals. So we've got the feeders and the lymphocytes from the vaccinated mouse have been fused to the myeloma, the Fox and the Fox NY cells. And what we're going to do is just incubate them overnight, 37 degrees, and let them grow for a bit, and then we'll apply selection tomorrow, and that will be a happy do, which has adenine, hypoxanthine, aminopterin, and thymidine in it, and that should select against the hybridoma, the hybridoma fusion partner, the myeloma, and select for the hybridomas. And the B cells and T cells that are down there, macrophages, they'll just die on their own by apoptosis. And in about a week, maybe 10 days, we'll have our first clones and we'll have to stop screening. Thank you. Rightio, so it's day two of the hybridoma protocol, and we've got to start selecting our hybridomas. Now, the problem is that we have got a myeloma in there, the FOXNY cell, which is immortal and will grow forever and overwhelm the culture unless we kill it, okay? Now, B cells and hybrids should be resistant to the selective protocol that we're going to use, and we want something that specifically kills the myeloma. So what we're going to use today is AHAT medium. So it contains adenine, hypoxanthine, aminopterin, and thymidine. So the fact you've got aminopterin in there is a folate antagonist that inhibits dihydrofolate reductase. So it means nobody can make either T or A or G from scratch, okay? Because they all require dihydrofolate reductase and tetrahydrofolate. Right here. So you have to rely on your scavenger pathway, and that relies on HBRT, hypoxanthine guanine phosphoribosyltransferase transferase. Now the myeloma is negative for that. It doesn't have it. Okay, so the FOXNY cells don't have that. Okay, so they're really susceptible to blockade by aminopterin. Okay. And so what we're doing is we're supplying thymidine and hypoxanthine. So thymidine will just be thymidine, and the cell will take it up. Hypoxanthine can be scavenged and made into either G or A. And we add A, so the only thing that the cell can't make is G. Now the myeloma can't make G, and so will die. But the B cells can make their G from the salvage pathway, the scavenger pathway. And any fusion between a myeloma and a B cell will also have that ability. They'll be able to use the salvage pathway and hence under these conditions survive. Now the B cells and the T cells and the macrophages in culture will die by apoptosis. Okay. So the only thing that should survive is your hybrid. Okay. And so after a few days of selection in this, we'll have killed off all the myeloma. Okay. And that's what we want it for. So after we've done that, after maybe 2 or 3 days, we'll stop using this and we'll just use HT medium, okay, without the aminopterin in it. So what we're going to do is we're going to feed these guys right now with 100 microliters of AHAT, which will go into the complete medium, and we'll start to kill the myeloma cells, the FOXNY unfused cells. The fused cells will survive, and the B cells and the macrophages and the T cells will all die, just naturally by apoptotic cell death. Right here. So let's get on and do that. So what I'm going to do is aliquot out some AHAT. Let's get on this. just bang through these. Now the important thing here is I don't want to use up too many tips, so I will reuse them from well to well. And then hopefully there's no cross-contamination. So there's 100. Just like that. So I've got a bit of work to do. I've got to fill in about 1,000 wells. And that's okay. It's worth it. Now, all that'll be left in a couple of weeks' time will be growing hybridomas. And so these will be immortalized B cells. And some of them will have the ability to make immunoglobulin. And hopefully we have some that make immunoglobulin that's specific to the top domain of polycystin 2. And so of course these clones are really what we're after, and we hope that we get a good antibody that can just churn out, a good hybridoma that can just churn out lots and lots of that one really good antibody. Of course they can be commercially viable. I have one good antibody which we sell through commercial vendors, and every Christmas we get a nice check that pays for mobile phones for all the kids, you know, so it can be quite a bit of money. Yeah, so I'll just go through this, and so I'll just continue doing this, and you can close down. Hi there. So now we're on day 4, and we've got 12 plates, and we have to feed them with more AHAP medium. So that's adenine, hypoxanthine, aminopterin, thymidine. And so what we're going to do is take 100 microliters off each and discard it, just to the top 100 microliters. And then we're going to fill up with another 100 microliters of AHAP medium. So for that, we're going to need sterile tips. Set your pipette at 100, and then we'll gently take 100 off the top, reusing the same tips, and then we'll replace with 100. So let's do, I'll just do one plate just to show you how. As sterile as possible. Just go 100. Approximately 100 out from each. Now it might well be that you cross-contaminate with clones, and another issue is you might spread infection from well to well, but it's the only way to do it really. I'm going through a lot of tips. But even if you do cross-contaminate, we're going to subclone, and we should be able to get down to just one clone in the end from each well. So, just like that. And then what you do is, like the last time, we aliquot out some AHAP medium and then just fill up the wells with 100 each. Okay? And that's that. Okay, thank you. Hi. So it's day 7 and we've got clones growing, and about 50% of the wells have got something growing in there. Now at the moment the cells are in AHAT, so that's adenine, hypoxanthine, aminopterin, thymidine. So the aminopterin has killed, we hope, most of the myeloma cells. So the only things that are surviving in there will be some of the feeder cells with the hybridomas, and you can see the hybridomas are growing up from the pictures I just showed. So now what we're going to do is we're going to dilute out the aminopterin. So what we're going to do is take 100 microliters of the top of each well and put in 100 microliters of AHT, okay, without the aminopterin. So it's got adenine, hypoxanthine, and thymidine. Okay, so that will dilute down the aminopterin. And the cells will feel happier because they'll be able to use more of the de novo synthesis for nucleotides, as well as using the scavenging pathway for nucleotide biogenesis. So I'll get on and replace 100 microliters from each well of these 1,000 wells, and then replace with 100 microliters of AHAT. And then we'll have another look at the cells on day 10, and then repeat that protocol. Okay, and then next week, days roughly 11 to 14, we'll look at wells that have got lots of cells in them, and we'll actually screen them for antibody against polycystin 2 toctomate. So I'll go and get on with this, and I'll see you again on day 10. Hi, so we're now on day 10, the day before we start screening, and we're going to feed the cells with AHT, which is adenine, hypoxanthine, and thymidine, and we're going to replace 100 microliters of medium with 100 microliters of this. Now I want to show you what the plates look like. Here we have a plate, and you can see there's 96 samples in here, and about 16 of them are going kind of yellow, and we'll probably pick those tomorrow. We'll probably pick those guys tomorrow. But at the moment we have to feed them, because over the next 4 days while we're selecting clones and assaying them, the clones will have to grow. And so if we replace half the medium right now, it will give them a bit of leeway to do that. Okay? So I'll get going to do that, and I'll see you tomorrow. Hi there. Okay, it's day 11 and we start screening today. So I'm just taking out 3 plates at a time, and what we're going to do is the ones that have gone yellow, we're going to harvest and put into a separate plate, which will be numbered, and then we'll have a sheet, and we'll put the coordinates of these plates onto this sheet, okay, and they'll map to a position on this second 96 well plate. Then we'll take them and get them assayed. We'll try to do that in 1 day. In fact, we'll have to do it in 1 day. And then at the end of the day, we'll try to rescue the clones that are yellow and are positive. Okay. So we'll just concentrate on the yellow ones first. So if you look at this plate, you can see some of the wells have gone kind of yellow. And those are the ones we want to assay quickly. If you don't do it quickly, they'll die on you, okay? So you want them as soon as they turn yellow. Okay, so we're going to do that. So what I'll do is I'll harvest, and then I'll call out to Tommy, who will write the coordinates on this new master plate, and then we'll take it and get it assayed. Okay, thank you. Right, so now we've got about 200 clones we want to screen. And so what we're going to do is we're going to use a high throughput Western blotting technique, where we run a Western blot with just one huge big well, and into that we put recombinant PC2 tap. So the tap is a tandem affinity tag. It's got flag tags and strip tags, and we just run it in this direction down through the acrylamide, so that you get basically 2 bands, low molecular weight band and high molecular weight band from the PC2. This is then transferred across to nitrocellulose, and then we use a cassette like this to basically probe to run multiple kind of Westerns across this. So as you can see on the inside, there are these grooves, and there's 27 of them. Okay, and what we do is we kind of sandwich our Western block, our nitrocellulose, between the two. Okay, and then we screw them together and then apply antibody into each of the wells. We put a control in the first and then apply antibody. And so each Western block can look at 24, 25 different supernatants. Okay, so to do 200 you're going to need 8 blocks. So if you come over here, we've actually got these set up. So first of all, there's a piece of kind of plastic, a sponge. It's under the membrane, and then you grab your membrane. I'm just going to use my fingers here. It doesn't matter. There's nothing toxic in there. And then it goes like this, and then it goes like this, and then we've got to number them. So we're up to 4 and 5 and 6 here, I think. So this will be 5, and this will be 6, yeah?
Yep.
So we'll just go 5. And then we go 6. Like that. And this goes on top, like so. And then we want to screw these in, have them nice and tight. And as you can see, each groove is now interfaced with the nitrocellulose, and so the protein is run down here as a big band across in this direction, and we're probing in this direction. So we'll see that in a second. Screw them all down. Then we've got to put in a control. So this is a control. We're just going to use 100 microliters of 1 on 1,000 flag, because of course our antigen is flag tagged. And I'm just going to wait a minute. Yeah.
You'll have enough, you just can't leave the space.
Actually, let's just adjust it. Okay, hold on a second. We're going to adjust this a little bit. If this happens, sometimes you miss, and you just got to adjust the position of the membrane. That's okay. Better to do it before you get started, of course. Otherwise you ruin everything. So let's just do this. I'm just going to pull this across. Sometimes it'll just slip. Yeah, here we go. Just like that. Yeah, there we go. That's better. Just that this groove here wasn't really in contact with the membrane. And we want it to be in contact. Oops, and something wrong here. It's just not. Something not quite engaged in. Sometimes this happens. Just got to get it. Sometimes it's screw it on again. So yeah, so just to show that it's a bit of a guddle, but you've got to get it right. So we first put on a control antibody in the very first lane. Kind of orientates things for us. So antibody. I'm just going to slot this along here. It should see the flag. So it goes into a hole like this, and then you squirt. And then again, and squirt. Okay, so that's your control. And then we'll get started on these guys. So you're doing this plate here, yeah?
Yep.
Okay, so I'll just start. So one tip every time. And we're going to go D1. And that will go here, you see, and it's red, and you can see it going in. I'll do another one because we missed that one. So as you can see, the medium is in contact with the big strip of the gel, under which is the polycystin 2. And it should bind to it. Well, if it's a positive, it'll bind to it. If it's a negative, there'll be nothing there. In fact, it might be quite dirty, but we'll be able to see the lanes because we won't wash too hard. There'll be a little bit of dirt there, and so we'll be able to count the lanes across until we get to our positive clot. So you just keep going like that. I'll do a couple more just to show you. So it fits in, and then you just fill it up and stop. Yeah. And a little more. Here we go. Cool. There we go. And that's us. We just keep doing that, and then we should be able to probe all 200 today. And if they're positives, we'll go right back there and we'll supplement them. Okay. We're going to wash the antibodies up out of these channels, and then rescue the blots and wash them 3 times. So you have a sucker like this, which just fits on the top of the wells like this, and you just got to get it to fit right, and it sucks it through. As you can see, we're sucking liquid from here through the grooves and out. So that cleans it up quite nicely, a little bit, and then we do the next one. And that's all it takes. And we're done. Now we've got to then just rescue these and put them into a TBS tween and do a Western blot basically. Just a classical Western blot, just like we did with the polycystin 1, 7E12. Goes out like this. And we rescue these. All we have to do is put these into containers. So we've got a couple of containers here, one here, one here, and we'll stick some of this. Quick rinse. Okay, so 3 5-minute washes, and then we'll put the secondary on. And it's just a classical Western blot after that. So that's one done. We'll just do another just to show you how it's done. Okay, bring in one up here. And there we go. One done. So we're doing 24 Westerns at the same time, really. 25 if you see the control. And we're done. Easy. Right, and then you've got to remember to tidy up at the end, otherwise people will be upset with you. It's very simple. So that was just after a half-hour incubation. Yeah. So we'll know in about an hour or two whether we've got monoclonals that recognise polycystin 2. And we should be right. Nice. I'll get these on shaking, and then Tommy will do the rest, and we'll have 8 Westerns to look at, but we can look at them very, very quickly. And all I've got to do now is just basically tidy up. Okay.